Today's video is on regulation of gene expression. So first we're going to look at the three ways in which gene expression can be regulated that you'll need to know about. And the first is controlling transcription. So this is choosing which genes will be transcribed. And transcribed means the synthesis of mRNA strands that can be translated at the ribosomes into proteins. Then there is controlling translation. So this is choosing which mRNA strands make it to the ribosomes to be translated into proteins. And epigenetics, which is chemical tags on histones or DNA that change the gene accessibility. But we'll be only looking at controlling transcription and controlling translation in today's video. So first is controlling transcription, and we're going to need to know two keywords for this section. The first is promoter region. So this is a specific sequence of bases upstream of a gene that controls gene expression. So we can see in this diagram here, we have a specific sequence of bases upstream of the gene. So this means further in this direction of the gene, so it's further up from the gene, it's not downstream. So downstream is the direction in which the gene is transcribed, so this is in this direction. So our promoter region is upstream of the gene because it's not in the direction of transcription. Downstream of the gene would be down here. Then there are transcription factors. So these are proteins that bind to specific promoter regions, and we'll see their uses now. So the controlling transcription stages. So first, in the nucleus, because this is where our transcription occurs, transcription factors bind to a specific promoter region. So we have our promoter region here, so we would have transcription factors binding to this specific DNA sequence, and more than one transcription factor can bind to this promoter region. And these transcription factors that bind to our specific promoter region recruit RNA polymerase. So we can see here that our RNA polymerase has binded to our transcription factors on our promoter region. So this is important because our RNA polymerase is very big. So this means that it's big enough to overlap the start codon of our gene. So you can see in this diagram, our transcribed region, so our gene is here, and our start codon is overlapped by this RNA polymerase that has bonded to these transcription factors. So this allows the RNA polymerase to bind to our DNA, to bind to our gene that we need to transcribe, and it starts the transcription of the gene. So this is very important because RNA polymerase doesn't bind to DNA alone. It needs to bind to the transcription factors first. So our transcription factors binding to our promoter region allows RNA polymerase to bind to the transcription factors, allowing our RNA polymerase to start transcribing our gene because it overlaps the start codon, allowing the RNA polymerase to bind to our DNA. So our transcription factors are produced when certain triggers ask for a gene to be turned on. So this could be hormones, for example. So when our body needs a certain gene to be produced, transcription factors for these promoter regions that express these genes will be produced so they can bind to these promoter regions, recruit this RNA polymerase to start the transcription of our genes to get mRNA. So now we're going to have a look at the example for this. So this is estrogen. So estrogen is a steroid hormone, so this means that it's made of lipids. And when a gene is turned off, this means that the binding site of the transcription factor isn't active. And when we say that this binding site isn't active, we mean it's either blocked or inaccessible. So this could be that our binding site on our transcription factor, so the DNA binding site, is curled in, so it cannot, it's not accessible to be able to bind to our promoter region, so it's inactive. And our hormones can activate these DNA binding sites on transcription factors. And this allows hormones to be able to turn genes on. So now we're going to have a look at how estrogen controls gene expression in this way. So because estrogen is a steroid hormone, this means that it's lipid soluble, because steroid hormone is a lipid-based hormone. So because estrogen is lipid soluble, this means it diffuses into the cell through the phospholipid bilayer via simple diffusion. So no proteins are needed, it's not facilitated diffusion because our estrogen is lipid soluble, it can easily just diffuse through our cell membrane into the cytoplasm. So once our estrogen is in the cytoplasm, it binds to a complementary receptor site on an inactive transcription factor. So this means that our transcription factor's DNA binding site is inaccessible. So our estrogen will bind to a specific complementary receptor site on our transcription factor. So it's not on the DNA binding site, it's on a completely different site. So this is our estrogen receptor site on our transcription factor that it binds to. And once our estrogen has binded to this receptor site on our transcription factor, this changes the shape of our transcription factor. And this is how it activates the DNA binding site. So you can see here, before our estrogen has binded to this transcription factor, this DNA binding site is curled in, so it cannot bind to the DNA. But once our estrogen has binded to our receptor site, this DNA binding site has opened up. It's become accessible to the DNA, so it is able to bind to our promoter region now. So our transcription factor, now activated by the binding of estrogen, enters the nucleus via the nuclear pores. So you can see that here. So they enter through the nuclear pores and they bind to specific DNA sequences or promoters. So you can see here that our DNA binding site has now bound to a promoter region. So a transcription factor has now been bound to our DNA. So this combination of transcriptional factors and DNA, so our transcription factors binding to our DNA, stimulates gene transcription by recruiting RNA polymerase, like we just saw in the previous slide. So this means that estrogen controls gene expression because it opens up our transcription factor by binding to a receptor site on our transcription factor, opening it up, allowing our DNA binding site on our transcription factor to be able to bind to our DNA to recruit these RNA polymerases to start gene transcription. So now we're going to move on to controlling translation. And the key terms here are RNA interference. So this is breaking mRNA before it can be translated. So this is important. So mRNA comes to the ribosomes to be translated into proteins, and by controlling translation, RNA interference chooses which mRNA are broken before they can reach the ribosomes to be translated into these proteins. Then there's endogenous, so this is naturally produced by the cell, and we'll see why this is an important definition in a minute. And there is exogenous, so this is man-made. So now we're going to have a look at the process of controlling translation. So first we start with double-stranded RNA. And so we can have proper double-stranded RNA, so this is endogenous, so you can see that here, and this is produced naturally by the cell, or you can have hairpin RNA, so this is exogenous, so this is man-made, so this is an RNA strand that's been folded around to make it double-stranded. It's not naturally double-stranded and produced in the cell. So our dicer enzyme now cuts the double-stranded RNA into smaller sections. And these smaller sections are called siRNA. So our dicer enzyme comes to either our endogenous double-stranded RNA or our exogenous hairpin, and this dicer enzyme cuts our RNA, our double-stranded RNA, into smaller sections called siRNA. And siRNA stands for small interfering RNA. And these are 20 to 25 base pairs, so these are very small. And one strand of our siRNA, so our smaller sections of double-stranded RNA, one strand is destroyed, and the remaining single strand binds with proteins to form a protein enzyme complex called RISC. So you can see here that our siRNA is now one strand. So one of our strands has been destroyed and the other is combined with proteins to form this enzyme protein complex called RISC. And RISC stands for RNA-induced silencing complex. So the unpaired RNA on our RISC complex binds to a complementary mRNA in the cytoplasm. So this mRNA has been transcribed from the DNA in the nucleus, it's come out of the nuclear pores, and it is coming to be translated at a ribosome. So this RNA on our RISC complex binds to the mRNA before it reaches a ribosome. And the enzyme complex RISC cuts this mRNA. So you can see here that our RNA on our RISC complex has bound to an mRNA before it reached a ribosome, and this RISC complex has cut our mRNA. And this silences the mRNA. So it cannot be translated now, it's been cut in half. So this means that the protein cannot be made, it cannot be translated at a ribosome. So this is how controlling translation works. So our double-stranded RNA is cut into siRNA by dicer, and a single strand from our siRNA combines with these proteins to form RISC, our RNA-induced silencing complex, and this RNA single strand on RISC binds to an mRNA, and this allows RISC to cut this mRNA, silencing it, not allowing it to be translated at a ribosome. So this is how translation is controlled. This is how gene expression is controlled, because the mRNA produced by the nucleus cannot be translated at ribosomes. And what are other applications of siRNA? So a quick recap from the previous slide, siRNA is small interfering RNA. So this is the smaller sections of our double-stranded RNA that is being cut by our dicer protein. And this siRNA is what helps with RNA interference. So this is cutting mRNA before it can reach the ribosome. So what are our applications of siRNA? So the first one is that we can block translations of genes to work out the gene function. And this works by, we can see the side effects when this gene is turned off. So when we turn a gene off, we can know the gene's function by viewing the side effects of when this gene is not on. And also, another application is that siRNA can be used to block the expression of genes that cause disease.